Okay, so for photosynthesis, we were looking at the light reactions, and remember that the light reactions happen in the chloroplast, and then you have these little discs here. One of these little discs is called a what? Thylakoid, right? And so each one of these are thylakoids. The whole stack is called granum. And this is where the light reactions occur. And when we look at this picture here, this picture, remember, I had drawn, I think, the lipid bilayer to show you that these photosystems are within the membrane of the thylakoid. And so this is what we're looking at here. So there's your big thylakoid here. And these are your photosystems inside. And so this picture right here is of the light reactions. What's the purpose of the light reactions? It's to make two things. Somebody is going to give me one thing that's made in the light reactions, which is really kind of the purpose of it.
NADPH.
NADPH. So NADPH, and then somebody else give me something different. Abby?
ATP.
ATP. So we have those two things that are made in the light reactions. Can somebody else tell me why we need to make those in the light reactions?
So you have to take the hydrogen off the water that's going through the light cycle to go into the Kelvin cycle to transfer carbon dioxide into glucose.
That's right. So these guys here, so you're right that the H's and the electrons in the H are the H's in NADPH. And the second step is the Kelvin cycle, which takes CO2, and then with the help of the ATP and NADPH, make your glucose, which is, remember, this is glucose, is the main thing we want to get out of photosynthesis. So this is what we want to get to. So the light reactions make these two things that are used in the part of the photosynthesis that actually makes the glucose. So we have these two different photosystems that both have chlorophyll A in them. And so the chlorophyll A, when the light hits, so this is, remember, the photosystem has the accessory pigments in the light harvesting complexes around the reaction center, which has chlorophyll A and your primary electron acceptor. So the light hits, gets transferred from molecule to molecule, eventually ending up as chlorophyll with chlorophyll A in both of these photosystems. Then remember, there's just slightly different chlorophyll A's because they absorb slightly different wavelengths of light. Then the electrons get excited. I'm going to put here a 2 in front of these. There are 2 electrons that get excited, and they never drop back down to chlorophyll A because the primary electron acceptor now takes these 2 electrons. So these 2 electrons, the 2 electrons that were once part of the chlorophyll, are now missing. So this is missing 2 electrons, this is missing 2 electrons. So we replace them. So therefore the electrons here are replaced by the electrons in water. So H2O, here's H2O. The water is split, so the oxygen part of the water leaves here, and so that's the oxygen part of the water. This is a half of an O2 is an O, all right, so that's the O from the water, and this is going to go into the air. This is the oxygen that the plant gives off. Then remember that the H's have the positive proton and the electron spinning around it. So the 2 electrons here from the 2 H's, so these are from H, from the 2 H's. Here, the 2 electrons. That means if we take the electrons, and these electrons are going to replace the 2 electrons that got excited, then we're left with 2 H pluses. So therefore, that's what these guys are from. So the 2 H pluses are the H's without the electrons, because the electrons go to replace these. Then these two, this chlorophyll A, these 2 electrons got excited, so this one's missing electrons. So what replaces them is the electrons up here. So these 2 electrons go through the electron transport chain. Why would they go through the electron transport chain? It's because each molecule, like why would the electrons leave the primary electron acceptor and go to all of these?
Even more electronegative.
That's right, so they're more electronegative. So as we go this way, we have increasing electronegativity. And so that's why the electrons go through the electron transport chain. The electron transport chain is a series of redox reactions. So when the electrons go to this molecule, this one gets reduced, this one gets oxidized. They go to this one gets reduced, this one gets oxidized, and so on and so forth. That's that releases energy. That energy is used to make ATP. So the electron transport chain is involved in making ATP. So then the electrons go and replace the 2 electrons in the chlorophyll here that got excited. So then the last thing that we need to look at is these 2 electrons. These 2 electrons that are once part of this chlorophyll here that are excited go through a small electron transport chain. And so here they are, the 2 electrons here. And they're going through those just like the reason here for the more electronegative. And they end up in a molecule of NADPH. And so notice that this box here, this box is not a reactant here. Look at it, ends in ACE, so what do we know about it?
It's an enzyme.
It's an enzyme. So it's the enzyme that catalyzes the reaction of putting these 2 electrons with NADP plus and the H pluses to make NADPH. All right, so the overall big picture here is what we want is we got ATP and the NADPH, and these 2 things we want because these 2 things are going to go to where? And be used where? In the Calvin cycle. So you're going to go to the Calvin cycle and be used. Okay. So when we look at this next picture is an analogy of the light reactions. Cute little picture, you have these little workers and so on. And it labels some things, and it doesn't label other things. And so what I'd like you to do is take a look at this picture, and what I'd like you to do is label what in this picture would represent the electron transport chain, what in this picture would represent chlorophyll A, what would represent the primary electron acceptor, and what would represent light. Alright, so take a minute and look at that picture and see, just literally try and label the parts on the picture.
I thought it was too.
The scale.
Teeter totter.
So, so let's start kind of at,
I thought they were just pushing.
let's look at light, alright, so, the word on here that should give you a clue. Photon. So, I chose this because I want you to correlate photon with light. And so, the people with the hammer, alright, that's the photon, that's the energy source coming into this system. And so you have here and here, this is the light coming in. Alright, so in real life in photosynthesis, what absorbs the light energy? The pigment. And eventually all that light energy ends up with what pigment molecule? So gets transferred the light energy to chlorophyll A. So what represents chlorophyll A here? Yeah, it's the little teeter-totters here. So this would be chlorophyll A. Now notice here, this represents the electron. So before the light hits it, the electron was sitting at the end of the teeter-totter. All right, and so that's here. So the electron was part of the teeter-totter. So that's chlorophyll A. The electron is part of chlorophyll A. Light hits it. This represents the electron becoming what? Excited. So here are your excited electrons. They don't fall back down to the teeter-totter or go back to chlorophyll A. These guys up here represent what? The primary electron acceptor that accepts those electrons. These electrons end up with NADPH. This electron slides down here and goes to this chlorophyll A, and it'll sit at the end of this teeter-totter. In the meantime, it spins this wheel, which is releasing energy to make ATP. So this represents the electron transport chain, all right? And so that's that. Okay, so that is your light reactions. We're going to skip a little bit. We're going to come back to this because this will make more sense after we do the Calvin cycle. We're going to skip all the way, we're going to skip quite a few right now, until I'm going to look at this picture. Keep flipping until you come across that. Okay, so let's look at this picture. This picture is showing you what we need to look at is how that electron transport chain makes ATP. So what is it about it? And give you the kind of the big picture of what's going on. And so here are your two photosystems, and you have your electron transport chain in the center here. And so this picture here is this picture here. So they're showing you, here's your lipid bilayer, here's your two photosystems, and so this is the electron transport chain. So light comes in, gets transferred from pigment molecule to pigment molecule. This little guy here is your what molecule? Right here, the center of these photosystems. What would this be? This is chlorophyll A. The arrow going up represents the electrons being excited, and this molecule is primary electron acceptor. These electrons go through the electron transport chain, replace these electrons. These electrons go to NADPH, and water replaces the electrons here. All right, so that's the same kind of, this is the same picture as the last picture, except it's showing you embedded in the thylakoid membrane. So this pathway here, this is the pathway of the electrons. So we have two electrons that replace the two electrons that get excited, and these guys move through the electron transport chain and replace these two electrons, and these guys eventually end up as NADPH. All right, so that's your path of electrons. So now, why is the path of electrons important? All right, so what happens as these electrons move through this electron transport chain? The source of the electrons is ultimately from water. So I want to point out here that, just like we talked about before, here's your H2O, 2 electrons are going to go through the electron transport chain. Notice here, there's 2 H pluses that gave off those 2 electrons, and a half of an O2, just like the last picture, that's going to go into the air. I want to point this out here because the H pluses are going to be important in just a second here, all right, from the splitting of water. So then the electrons, when they go through the electron transport chain, very similarly to the electron transport chain in the mitochondria, when the electrons go through the electron transport chain, as I said, it's a series of redox reactions which releases energy, and that energy can be used to pump H pluses into the thylakoid. So when I say the word pump, that means that it's active transport. It requires energy. So I'm going to write active transport here. Why is it active transport? Because, look at here, low H plus concentration outside the thylakoid, high H pluses in the thylakoid. So it's going from low to high, which requires energy. The energy is not in the form of ATP. It's not using ATP for energy. It's using the energy of the redox reactions of the flow of electrons through the electron transport chain. So these H pluses are pumped, and so now we have even more H pluses in the interior of the thylakoid, which is called the thylakoid space. So that creates a really high concentration of H pluses within the thylakoid. All right, that's the purpose of the electron transport chain. So now, why do we want H pluses in the thylakoid? Well, remember that we said that the electron transport chain makes ATP. So how does the flow of those electrons do with making ATP? Because when they pump in the H pluses into the thylakoid space, making a really high concentration of them inside, there's another protein, our old friend ATP synthase. All right, ATP synthase, remember, is the enzyme from cellular respiration that is involved in making ATP. So ATP synthase, normally just hanging out in the membrane does not work. What has to happen in order for ATP synthase to work?
Diffusion.
The diffusion of the H pluses. All right, why doesn't it work? What does the diffusion of the H pluses do to it?
Changes shape.
Changes its shape so that ADP and the phosphate can fit to the active site and make ATP. And so that's the only way that occurs. So therefore, it's really important to have a very high concentration of H pluses in this thylakoid space for that enzyme to work to be able to make your ATP. And so the electron transport chain is instrumental in that. So ATP synthase changes shape. I'll write here, changes shape when H plus diffuses through it. Allowing ADP and the phosphate to bind to the active site. To make ATP. So if the ATP or the H plus doesn't diffuse through, then no ATP is going to be made. And if no ATP is made, then we have no Calvin cycle occurring. Yes.
Could we also call that chemiosmosis?
Absolutely. All right. So this right here is chemiosmosis. That's right. And that is the same thing that really was occurring in the mitochondria. Like one step ahead of me because that's what I was going to talk about next. All right, so if we look here, then what we've got going on is ATP made, NADPH made, and they're going to go to the Calvin cycle. Where does the Calvin cycle take place in the chloroplast? So here's your chloroplast of the cell. Calvin cycle takes place out here. What is out here? Stroma. The stroma is the liquid in the chloroplast. And so notice here that ATP and NADPH are not made within the thylakoid. We use the thylakoid, but they're actually made on the outside of the thylakoid. Why is that beneficial? Why is that beneficial?
Okay, so it doesn't have to go through the thylakoid?
Yeah, absolutely. It doesn't have to go through the thylakoid, so if it's made inside, it'd have to somehow be transported out. And so now, since they're made on the outside with the help of the thylakoid, they're right where they need to be, which is in the stroma, all right? And so that's a beneficial thing. Do you have a question? Sorry.
Yeah, I was wondering if for every hydrogen that diffuses through ATP synthase, does that make one ATP or can it make multiple?
It can make multiple, but it's kind of like the same idea with the actual numbers of the end-of-cycle respiration. They don't really know for sure. All right, so we can. But what we'll talk about here is that these guys are made almost in a 1-to-1 ratio. Again, we're not sure exactly. So you'll see the different publishers will have different numbers there. All right. All right. So that is the light reactions. So now we're going to go back to the part that we skipped. But we're just going to go back to this one. All right? We're not going to go all the way back to where it says cyclic, because that we still need to understand that the best. You need to kind of understand the Kelvin cycle first. So go back to where it says a comparison of chemiosmosis in the mitochondria and the chloroplast. So chemiosmosis occurs in both places. Chloroplast and mitochondria both generate ATP by this process of chemiosmosis. The H plus is diffusing through this ATP synthase so that it's in the right shape, so that it can put the ADP and the phosphate together, which is what we just looked at. Oh, I wanted you to look at the second bullet and see if you could fill in the blanks. All right, so comparing what happens in the mitochondria versus the chloroplast. So in the mitochondria, got the wording on this. Energy starts out in food, so that's your glucose molecule, starting out in your food molecule, and ends up in what? So where's the transfer of energy occur from the beginning to the end of cellular respiration in the mitochondria? See if you can put what you would think it would go there. And the second part of that is what you think for photosynthesis. So chloroplasts transform light energy into the chemical energy of what? So what's the beginning of photosynthesis? The source of energy is light. At the end, where is that light energy now at? It's been transformed into another form of energy, chemical energy, in what molecules? So mitochondria, cellular respiration, what goes in is your food, so glucose is an example of that. And that energy that was stored in the bonds of the glucose molecule is at the end of cellular respiration in what molecules? ATP. And then for the chloroplast, for photosynthesis, you start out with light energy, and that light energy is transformed and eventually is in the bonds of what molecule? Glucose, absolutely. All right, so first is ATP, next is glucose. So then let's look at this picture here. This picture is showing you and comparing and contrasting this process of chemiosmosis, making ATP from the diffusion of H pluses through the ATP synthase, and looking at where that occurs. So how you look at this picture? So in the mitochondria, so they're talking about how we make ATP in cellular respiration, right? And we can assume that since they're talking about the mitochondria. And this is how we make ATP for photosynthesis. And so this box right here, when they show this box right here, this is showing you where it occurs, where we would find the electron transport chain in ATP synthase in the mitochondria. And over here, it's showing you where we would find it in the chloroplast, which is in the thylakoid membrane. All right, so how we look at this picture is when we're looking at and talking about the electron transport chain and ATP synthase for the mitochondria, we disregard these words over here, and then this is what's true about the mitochondria. So this, for the mitochondria, is this membrane here. Just this membrane here, what would we put in front of that? What is this membrane? It's a phospholipid bilayer here membrane, but where is it in the mitochondria? Like, what is the inner membrane of? It's the cristae, which is the inner membrane of the mitochondria. So I'm gonna write inner membrane or cristae here. And so remember that then you have the inner membrane space, which is the gray spots here, and then you have the matrix, which is right here. And so if you remember that when the electrons go through the electron transport chain, it pumps the H pluses into the inner membrane space, so you have a really high concentration here, so they diffuse back into the mitochondria and make ATP, all right, that way. So that's what happens in the mitochondria. Now when we look at it on this side, disregard these words here and just look at what happens in the chloroplast. The membrane is what? What kind of membrane is this? What membrane is it of? Like in the
thylakoid.
This is the thylakoid membrane. That's where we find this. And so looking at this, this is the inside, this is the thylakoid space, so this is inside the thylakoid. So inside thylakoid. This is out the stroma, which is outside the thylakoid. So now, when the electrons go through the electron transport chain, they pump the H pluses into the thylakoid. So the H pluses go in this direction into the thylakoid, all right, and then diffuse back out through ATP synthase into the stroma. All right, so that's just comparing and contrasting. So ATP is made in both of them, but the ATP in the chloroplast is made solely for the Calvin cycle. So so photosynthesis doesn't need any help from cellular respiration to run. It makes its own energy or its own ATP. So then cellular respiration makes the ATP for all the other things going on in the cell. All right, and so that's the relationship between them. Okay. So underneath this picture, you have some bullets here, and so. The second bullet there says water is split by photosystem blank on the side of the membrane facing the blank. For the next 3 bullets, there's 2 at the bottom of this page and 1 at the top. I'm gonna see from our discussions, you should be able to fill in those blanks. See if you can fill in those blanks.
Okay. So that would be photosystem 2, right? Yep. Okay, now that's what I don't know. The side of the membrane facing. Oh, wouldn't it be the out? How do you say outside? Okay, because it gets the light. So facing the stroma. Yeah. We're supposed to do the next 3. So the diffusion of hydrogen ions from the blank back to the blank. Powers ATP synthesis. Yep.
Yeah, from back in
Is that what it's called? Thylakoid space?
Yeah.
Okay. Space. Like the inside of the thylakoid. Then ATP and NADPH are produced on the side facing the stroma. Yes. Where's the blank? Oh, is that where the Calvin cycle takes place? Are you talking about Brave New World? Were you thinking about stoma or stoma? Yeah. Gosh. Ugh. In my LA class, we're reading one like it now. It's like a another dystopian novel where all the women are just like producers. Oh my god. Yeah. It's pretty good actually. We just finished it.
Sounds interesting.
It's better than Shakespeare. I don't like Shakespeare.
I can read like the Shakespeare picture novels, not the actual novel.
Yeah, I do not like it. Do you like Shakespeare?
I think the stories themselves are interesting, like you know them, but like trying to read the book sometimes is
No.
yeah.
Yeah. There was, I read Taming of the Shrew, and I guess there's a ballet of Taming of the Shrew.
Yeah, I have a
No, it's not type of bird.
shrew.
Alright, let's go over this.
Well, a shrew is a rodent too.
Alright, let's see what we got here. So water is split by photosystem 2
Okay, we got that.
on the side of the membrane facing the thylakoid space. I want to show you this real quick. Let's look at why that is. So here's the water. Water is split by photosystem at photosystem 2 at the very beginning, right?
Yeah.
And then when it's split, the H pluses landed on the side facing the thylakoid space because we need the H pluses to make our high concentration of H pluses in the thylakoid. Okay? And so that's what that means. All right, next bullet. The diffusion of H pluses from the thylakoid space back to the stroma powers ATP synthase. So it's going from the inside of the thylakoid to the outside. And then lastly, ATP and NADPH are produced on the side facing the stroma where the Calvin cycle takes place. So remember, it makes sense for them to be made on the outside of the thylakoid so that in the stroma, because that's where the Calvin cycle actually takes place. All right? And so, and that's the light reactions. All right? So now we need to look at how these guys are used with carbon dioxide in the Calvin cycle to ultimately get our glucose. So that's what we're going to look at next is the Calvin cycle. So the Calvin cycle uses both of those things. Calvin cycle, like the citric acid cycle from cellular respiration, regenerates its starting material after molecules enter and leave the cycle. So that means it's a cycle, meaning that you have a molecule here that reacts with CO2 that comes in, goes through a series of reactions, and you regenerate that same molecule that can then react with the next CO2. The cycle builds sugar from smaller molecules by using ATP and the reducing power of electrons carried by NADPH. Those two things are coming from the light reactions. The carbon that's going to be part of glucose enters as CO2. Then it says, it leaves as a sugar named glyceraldehyde 3-phosphate, or G3P. So let me explain the relationship of that and sugar, or this glucose. So when we say G3P, G3P is a sugar that is 3 carbons long. So therefore, you have this 3-carbon sugar. Glucose is how many carbons? 6, right? So basically it takes 2 of these 3-carbon molecules to make 1 glucose molecule, which is a 6-carbon molecule. So this is kind of a step before getting to glucose. And then for the net synthesis of 1 G3P, the cycle must take place 3 times, mixing 3 molecules of CO2. Fixing, this is referring to carbon fixation, and I'll talk in just a minute about that. Again, we talked a little bit about it at the beginning of the notes, but probably don't remember. So I'll talk in a minute about that. So let me talk about why it takes place 3 times this cycle. So at the end of the cycle, do you see how you get 3 carbons, a sugar that's 3 carbons big? Where does the carbon that is going to be part of the G3P and eventually part of glucose come from? It originates as the carbon coming in as CO2. So therefore, you see to get 1 G3P, that you would need 3 carbon dioxides to make your 3 carbon sugar molecule, all right? And so then ultimately we need 6 carbons then to get your glucose. So what happens is the cycle takes place 3 times because the CO2 enters one at a time. So there's one time, you get 1 carbon, so you have to have 3 turns of the Calvin cycle to get 1 G3P molecule. All right, so let's we're going to look at more specifically the carbon cycle, or the Calvin cycle here. And there are 3 phases. Carbon fixation, I'll talk about that in just a minute, but this is catalyzed by Rubisco. Catalyzed by Rubisco. What do you think Rubisco would be? This is an enzyme. Although it just doesn't follow the rules, it doesn't end in ACE, right? So this is an exception, but it's still an enzyme. Then there's the reduction phase. What does reduction mean? Gaining electrons. And then there's the regeneration of the CO2 acceptors, the molecule that originally reacts with the CO2 in the cycle. So we're going to look at all 3 of these phases here in this next picture. All right, so the Calvin cycle, just like the citric acid cycle, you don't need to know all the intermediates and stuff. We don't have to memorize the names of all these molecules here. But let's look at, here are your 3 phases that we just wrote here. Here's phase 1, here's phase 2, and here's phase 3. Let's just talk about what happens here. So at the top, you have 3 CO2s coming in. And notice it says 1 at a time. They're just showing you the big picture because it takes 3 CO2s to come in 3 turns of the Calvin cycle to get your 1 G3P here. So they kind of put 3 turns of the Calvin cycle all together in 1 to show your ultimate G3P molecule. So your CO2 comes in. Here's your first molecule, RUBP, reacts with CO2 to form goes through a chemical reaction and starts to form these intermediates in the Calvin cycle. Catalyzed, here's your enzyme by Rubisco, and so that's the enzyme there that catalyzes that reaction. So then, that's called carbon fixation. Carbon fixation is carbon from carbon dioxide incorporated into an organic molecule. And that's what just happened there. And Rubisco helps with that. So then, carbon from CO2 is now in these organic molecules, goes through a series of reactions. Notice here, we have ATP. And ATP is being used, right? So therefore, these reactions need some energy. Where did this ATP come from? Yeah, this is from the light reactions. Fly up here that keeps
Oh.
like looking around. Alright, so then so it uses some energy, and then now notice we're entering into phase 2, reduction. Reduction means gains electrons. Where's the electrons coming from? NADPH. So this carried the electrons from where? Low light reactions. So that's why this is reduced, because it is giving electrons to these intermediate molecules. That's the use of NADPH. So now look at the end, we get 6 G3P molecules. That's your 3 carbon sugar. Notice that 1 of those is gonna leave the cycle, and this is your 3 carbon sugar that, remember I said, can be used to make your glucose. So I'll put your times 2 to make your glucose. And so So that's where that comes out. Then the remaining G3Ps, these remaining 5, go through a series of chemical reactions. We call it the regeneration of CO2 acceptor, which is RUBP. So it goes through a series of reactions, that requires some ATP. The ATP came from the light reactions. And ultimately we get RUBP regenerated, so now they can react with the next CO2 and the cycle can continue on and on and on. All right? And that is your Calvin cycle and how it makes ultimately your glucose, G3P first and then ultimately your glucose. All right? So let's do some totals here. Let's do say needed to make 1 G3P. So if we total something, so what do we need? We need some ATP. So how many ATPs do we need? You need NADPH and you need CO2. So put some numbers in front of there. From the light reactions, depends on what source you want to look at.
So the numbers are
not So you should have for ATP, 6 ATP right here is used and 3 here. So that leaves us a total of 9 to make 1 G3P molecule. For NADPH is 6. Because remember, these are the totals of 3 terms, all right, to make 1 G3P. Okay, all right. So you don't have to multiply it by 3. All right, and then CO2 is 3 CO2s. So if I asked you the same thing for a glucose molecule, you would tell me what? What would I have to do with my numbers? Double. All right, so it'd be 18, 12, and 6 CO2s. And notice here the 6 CO2s, that's why we have 6 CO2s entering into the reaction.
Is it a coincidence that it's 3, 6, 9 and 6, 12, 18?
Yeah.
Okay.
what?
Okay.
Goes by 3s.
So let me ask you this. So then looking at what's made here, let's go back, and you don't have to go back in your notes, but I just want you to look at this picture here. So this is the picture right here that I want you to look at for a second. So in the light reactions, both of these are made in roughly equal quantities. We're going to say close to equal. I don't know for sure exactly, but it's close to equal. So NADPH and ATP are made equally, and therefore they go to the Calvin cycle. Now when we just talked about the Calvin cycle, are the ATP and NADPH used equally in the Calvin cycle? No, which one's used more? ATP, right? So imagine this, you've got this amount of ATP and this amount of NADPH, roughly equal, going to the Calvin cycle. Go through the Calvin cycle, which one am I going to run out of first? I'm going to run out of ATP because I use it more, or the plant uses it more. All right, and so what you're going to end up with is some NADPH hanging around, and it can't do the Calvin cycle because the Calvin cycle ran out of ATP. So what the plant does to do this is, in the light, when that happens, when they start to run out of ATP to do the Calvin cycle because it's used up more, they go back to the light reactions and only do the part of the light reactions that has to do with the electron transport chain. Because what does the electron transport chain do?
Drive.
It pumps the H pluses into the thylakoid so that you can make ATP. So to understand this, then we're going to go way back to the part that we skipped called cyclic electron flow, because that's what happens. And you wouldn't have understood the need for this until we talked about the Calvin cycle. So cyclic electron flow happens when ATP gets used up. So cyclic electron flow uses only photosystem 1 and produces only ATP. Find it, keep looking back. It's right before the chemiosmosis picture. Underneath, how about this, the analogy picture? That's a better one. Underneath the analogy picture. So cyclic electron flow uses only photosystem 1 and produces only ATP. Because that's what we need, because it's used more in the Calvin cycle. So this cyclic electron flow generates surplus ATP, satisfying the higher demand of ATP in the Calvin cycle. You need more ATP in the Calvin cycle. So this picture, let's look at this. Mine is colored. Yours is not, so this is gray area, and this is nice pretty colors. All right, that's because this is the part that's dealing with cyclic electron flow. So this is the part right here, all right, that's colored here, that's important. Yours doesn't show that, so I'm putting a box around it, so you can put a box around it because that's the important part here. So remember what normally happens. Here's your two photosystems. Light comes in, the energy gets transferred to chlorophyll A. Electrons get excited. Normally these electrons go through the electron transport chain, make ATP, and these electrons go to NADPH. In this case, we don't need any more NADPH. So we don't need any more because we already have enough. We're running out of ATP for the Calvin cycle. So this part is not going to happen. And so what happens when in cyclic electron flow is when these electrons get excited and go to the primary electron acceptor. Instead of it going to be made useful in making NADPH, it's going to go back and be dropped off in the electron transport chain. So then the electron transport chain happens, which causes H pluses to diffuse in and causes ATP synthase to make more ATP. So it goes through the electron transport chain, makes more ATP, goes back to chlorophyll A, gets excited, instead of going to make NADPH, gets dropped off in the electron transport chain, makes more ATP, gets excited. So we're making only ATP. So then what that's doing is, remember that these guys were made originally in equal quantities, but when we go to the electron transport chain, ATP starts to get used up. So then the plant back here in the light reactions starts just doing this part here, which only makes ATP, so it brings the ATP levels back up, and then guess what it does? Once the ATP levels are back up, it goes back to normal and produces both of them again. Does it make sense? Let's write this down. All right, so let's write it. I'm going to put a couple of steps here so that so we can write that the chloroplast of the Calvin cycle uses more what than what?
ATP. Then NADPH.
Yeah, uses more ATP than NADPH, which are made roughly in equal quantities. So what are we going to start running low on? ATP. So if ATP runs low, the NADPH is going to start accumulating because it's not going to be able to be used in the Calvin cycle because the Calvin cycle needs ATP. So this is going to stimulate cyclic electron flow. This stimulates cyclic electron flow which makes what but not what?
ATP, but not an ATP.
Which makes ATP but not NADPH. And so this, what does this allow? This allows your ATP numbers to kind of catch up with NADPH. To kind of catch up. With NADPH. And so once the ATP numbers come back up, it'll go back to non-cyclic electron flow and make both of them in equal quantities. And then as in the Kelvin cycle, it starts running out of ATP, it'll go through cyclic and make more ATP, and then go back to making both equally, and so on and so forth. And so that's what happens in cyclic electron flow. And that, my friends, is photosynthesis. The back page is really just a review. It just shows you a big picture of the chloroplast and, you know, the different stuff that goes in and out, and so on. So, real quickly, before you leave, I can stop this.